Hello friends, welcome back to my channel Plant Pathology Pathshala. So here is another video on plant virology. We have already discussed the nature, composition, architecture of plant virus. Then I have made a separate video on viroids. Then properties of virus we have already covered, variability. Now in this video we will discuss about satellite virus and satellite RNA. So without further delay, let's start the video. So these are the slide we have already covered about the general characteristic, then the composition of virus, architecture of virus. Then I have also prepared a full video on viroid about the definition, structure, composition, architecture, location, movement and transmission, classification, then diagnostic procedure, and finally the management. Then properties of virus and variability, both generalized and specialized mechanisms. Next, the satellite agents. So, first the definition. So, what is a satellite agent? Why we call it as satellite agent, not the virus? What is the difference? So, the satellites are subviral nucleic acid that require a helper virus for replication, then encapsulation and movement, but share a little or no sequence homology with the helper virus. I think you people are well acquainted with this definition. Everybody knows that the satellite agent means this require a helper virus for replication, encapsulation and movement. But this important point generally students used to forget about the sequence homology. There should not be any sequence homology with the helper virus. The satellite agents never show any sequence homology. There may be little sequence homology, but whole sequence homology will be not there with the helper virus. If any sequence homology is there, significant sequence homology is there, then that will be not called as satellite agent. They will be called as defective interfering RNAs. If it is a RNA, it will be called as defective interfering RNA, or if it is a DNA, it will be called as defective interfering DNA. Okay, so remember this fact. Next, about the classification. So 3 types of satellite agents are there based on its dependence on the helper virus. Up to how much extent they are depending on the helper virus, they are classified into these 3 groups. So the first group is the satellite viruses. So satellite virus actually possess their own coat protein or own protein or capsid, but only require the helper virus for the replication. They are structurally independent particles. But in case of satellite RNAs, they are very small RNA molecules which lack capsid formation, means they lack the protein coding capacity, so it can't synthesize in its own protein and fail to do the encapsulation. It depends on helper virus both for replication as well as encapsulation or packaging. Okay, so this is the main difference between satellite virus and the satellite RNAs. Its dependence on the helper virus. For replication, the satellite virus only depend on the helper virus for the replication, but the satellite RNA generally depend on helper virus for both replication as well as for encapsulation. Why this happen? Because the satellite virus are generally the nucleic acid is generally bigger, more than 1 kb. Okay, so it contains ORF that code for gene or the protein. But here in case of satellite RNAs, this genome size is very small. The RNA is very smaller. So no ORF is there. There will be no protein will be coded. Okay. Then example of satellite virus. This is the first satellite virus. STNV, satellite tobacco necrosis virus. And this was first actually discovered by Cassanis. The scientist name is Cassanis, and he discovered the virus in the satellite virus in the year 1962. Then about the satellite RNA. So satellite RNA generally, the example is satellite cucumber mosaic virus, satellite RNA of cucumber mosaic virus. And generally the satellite RNAs are reported, or it is commonly associated with CMV, I have given here, and another two virus generally they are associated, that is Nepo virus and the Sobebo virus. Nepo means the nematode transmitting virus, and some southern bean mosaic virus. So for these three viruses, CMV, Nepo and Sobebo, generally satellite RNAs are associated, commonly associated. And the first discovered satellite RNA is the satellite RNA of CMV. This was the first discovered satellite RNA, and it was discovered by Kapper and Waterworth in the year 1977. So the scientist Cassanis in the 1962 discovered the first satellite virus, that is STNV, and Kapper and Waterworth in the year 1977 first discovered the satellite RNA of CMV. Okay. Next about the DNA satellites. So here in this two conditions, satellite virus and satellite RNA, here only the RNA is the genetic material, but here the circular single stranded DNA is the genetic material for the satellite DNA. And satellite DNAs, they don't code for code protein. They are associated mainly with the begomovirus. So very few DNA satellites are there, and examples are alphasatellites and betasatellites. Okay. So this is about the classification of satellite agents. Next, a clear difference. So generally in the mains exam or in the MCQs also, it can be asked the difference between satellite virus and satellite RNA. So first about the protein code. So protein code is present in satellite viruses, whereas it is absent for the satellite RNA. Then protein coding gene, code protein gene, it is encoded by satellite genome, it is not encoded. Then structural nature, it is an independent particle, but this is naked RNA molecule. Then encapsulation, this is this can do or perform its self encapsulation, whereas the satellite RNA, it is encapsulated by helper virus. Then dependence on helper virus, mainly for the replication for satellite virus, whereas the satellite RNA depend on helper virus for replication as well as packaging. The nature, so satellite virus is subvirus like particle, but here the satellite RNA is naked subviral RNA. Example, STNB, or here satellite RNA of CMB. Who discovered this? I have already told you. So this is about the satellite RNA and satellite virus. Then the detail about the satellites of DNA satellites of plant viruses. So first the definition. So DNA satellites are small circular single stranded DNA, not double stranded. The DNA satellites are circular and single stranded DNA molecule associated mainly with the begomovirus, and they do not code for any code protein, and they are encapsulated by the helper virus. So the difference mainly the DNA virus and DNA satellites. The main difference is the DNA satellites can't synthesize its own code protein. They need a helper virus for encapsulation or for synthesis of the code protein. And two main types of DNA satellites are there, that are alpha satellite and beta satellite. So the alpha satellites, these are same only, the genome is same circular single stranded, but they are different based on the coding capacity. So alpha satellites generally encodes rep protein, replication associated protein. Rep protein, when we will discuss the virus replication, we will discuss these things. Rep protein, it is very essential for the replication of DNA viruses. So as it encodes the rep protein, it can perform its replication autonomously. So require helper virus only for movement and encapsulation. So usually associated with symptom modulation. So it modulates the symptom of helper virus. But in case of beta satellites, it doesn't code for rep protein or replication associated protein. If rep protein is absent, means it can't perform its replication autonomously. So it entirely dependent on helper virus for replication, movement and encapsulation. So major determinant of symptoms and severity and pathogenicity. So you can consider that like this alpha satellites are satellite viruses, and this beta satellites are similar, showing similar characteristic with satellite RNAs. Here also only the alpha satellite depend on helper virus only for the encapsulation, just like the satellite viruses, whereas the beta satellites depend wholly on the helper virus for replication, movement as well as encapsulation, just like the satellite RNA. The main difference lies on its coding capacity, its ability to code the rep protein, that is replication associated protein. Okay, then example, it is commonly associated with begomovirus. Begomovirus means bean golden mosaic virus. So they are generally transmitted by whitefly. Bemiciata becki is a major disease. Okay. Examples are tomato leaf curl disease, tomato leaf curl virus, DNA satellites of cotton leaf curl virus, and DNA satellite of tomato leaf curl virus. So here the quick revision point. So satellite virus own protein capsid it can synthesize, but it depend on helper virus for only for the replication. But satellite RNA, no capsid, it depend wholly on the helper virus for replication, movement, encapsulation, everything. And DNA satellite, generally two types are there, alpha satellite and beta satellite. Alpha satellite can perform its replication autonomously, whereas beta satellite depend wholly on the helper virus for replication, movement and encapsulation. So this is about the satellite agents. I hope everything is clear to you now. So if you have any query, please let me know in the comment section. I will try to clear your doubts. So I have created a separate WhatsApp group for sharing the videos as well as PDF notes, whatever I am sharing in the YouTube. So join the group by using the QR code given in the thumbnail. So this is all about today's video. So thank you for watching.